Good day, this is Professor Will, CBMD PhD. Today is Friday, July 16, 2021. It is 4:50 and 10 seconds p.m. Eastern Standard Time. In today's research, cancer cells. So let's hop to it. The single cancer cells have different appetites for fatty acids. Date July 16, 2021. Source, Institute for Systems Biology. A recently developed method provides new insights into cancer biology by allowing researchers to show how fatty acids are absorbed by single cells. Fatty acids, along with glucose and amino acids, are a major energy source for cellular growth and proliferation, and abnormal fatty acid metabolism is often seen in cancer. Researchers have been collaborating for years to develop a series of chemical probes and analytical approaches for quantifying cellular glucose uptake, lactate production, amino acid uptake, and other cancer-related metabolites. Unlike glucose and amino acids, however, the mechanisms underlying the uptake of fatty acids in the cells have been lesser known and difficult to discern. The technical tools for measuring fatty acid uptake at a single cell level are extremely limited, and this work is the first example of profiling fatty acid uptake in conjunction with aberrant protein signaling in cancer cells at single-cell resolution, and represents an important advance in the single-cell metabolic assay. And to profile the fatty acid uptake, the researcher chose a surrogate molecule that was structurally similar to natural fatty acids. And this similarity tricked the cells into taking up these surrogates like the native ones. Then, using a unique dendrimer molecule, a tree-like polymer, the researchers achieved precise quantification of those surrogates from single cells. Applying this new single-cell tool to a brain cancer model, the researchers identified that fatty acid uptake was differentially regulated by two downstream effectors of the mammalian target of a rapamycin, mTOR, a critical regulator of cell proliferation and protein synthesis. The results revealed a compensatory activation of fatty acid metabolism upon oncogene inhibition or attenuation of glucose metabolism in these brain cancer cells, and uncovered a novel combination therapy that targets its bioenergetic flexibility to synergistically block the tumor growth. And this novel tool opens new avenues for studying how fatty acid metabolism affects biological systems. It has also inspired researchers to develop more metabolic probes for single-cell analysis. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.